The tumor suppressor protein p53 is often called the guardian of the genome, and for good reason. It sits at the crossroads of cell cycle control, DNA repair, apoptosis, senescence, and metabolism. One of its lesser-known but profoundly important roles is its influence on autophagy, the cellular recycling process that helps maintain homeostasis and protect against damage. The way p53 governs autophagy is intricate, context-dependent, and deeply tied to both aging and cancer. This duality makes p53 not just a gatekeeper against tumors, but a critical regulator of how cells survive or deteriorate as we age. Under normal physiological conditions, p53 helps maintain genomic integrity. When DNA damage or oxidative stress occurs, p53 is activated and initiates a range of protective responses. These include halting the cell cycle to allow for repair, triggering apoptosis if damage is beyond repair, or inducing autophagy as a salvage pathway. Through autophagy, cells can remove damaged mitochondria via mitophagy, clear toxic protein aggregates, and recycle nutrients to survive under stress. In this way, p53 indirectly supports cellular rejuvenation and metabolic balance, reducing the burden of senescence and suppressing the earliest steps of tumor formation. However, p53's role in autophagy is not unidirectional. It can both promote and inhibit autophagy depending on its localization and cellular context. Nuclear p53, which functions as a transcription factor, tends to activate autophagy by upregulating genes like DROM1, damage-regulated autophagy modulator, and AMPK, or by repressing mTOR signaling. This enhances the cell's ability to clean itself and cope with metabolic or genotoxic stress. But cytoplasmic p53 can actually inhibit autophagy by blocking AMPK and activating mTOR, both of which reduce autophagic flux. This creates a feedback loop. Under some conditions, p53 activation promotes repair and survival. Under others, it pushes the cell toward arrest, senescence, or death. This complexity becomes particularly important in the context of aging. As organisms age, low-level, chronic activation of p53, driven by ongoing DNA damage, telomere shortening, and mitochondrial dysfunction, can lead to persistent cell cycle arrest and the accumulation of senescent cells. These cells, in turn, secrete pro-inflammatory factors that damage tissues and fuel age-related diseases. In this environment, p53 may inadvertently promote aging by favoring senescence over autophagy. If autophagy were more robust, these damaged cells might instead be repaired or cleared efficiently. Thus, p53's delicate balancing act between protection and pathology can tip in the wrong direction as autophagic capacity declines with age. In cancer, the role of p53 is equally paradoxical. Loss of p53 function is one of the most common genetic alterations in human tumors. Without it, cells bypass key checkpoints, accumulate mutations, and evade apoptosis. This not only allows tumor initiation but may also disrupt proper autophagy regulation. In some cancers, reduced p53 function leads to insufficient autophagy, contributing to genomic instability. In others, tumors hijack residual autophagy to survive under nutrient-deprived, hypoxic conditions. Here, the absence of p53's fine-tuned control allows cancer cells to use autophagy as a survival mechanism rather than a tumor-suppressive one. This duality presents a warning. Manipulating p53 or autophagy independently without understanding their interdependence could backfire. For instance, activating autophagy in cells with mutated or absent p53 might not restore health. It could promote tumor growth by enhancing stress tolerance. Conversely, reactivating p53 in aged tissues could increase senescence if autophagy is too weak to clear the damaged load, resulting in greater inflammation and degeneration. Still, there's hope in therapeutic modulation. Drugs that restore p53 function, such as APR 246, or stabilize its activity in cancer cells, are under investigation. If combined with autophagy-enhancing agents, they may reestablish a more youthful, damage-clearing phenotype in cells, boosting regeneration and suppressing tumorigenesis at once. Similarly, targeting downstream p53, autophagy signaling like AMPK activation or mTOR inhibition, may offer safer routes to rejuvenation without triggering excessive senescence or apoptosis. Ultimately, p53's influence on autophagy tumor suppression, and aging highlights how tightly interconnected these biological processes are. It's not simply a matter of turning on or off a single gene or pathway, but rather of navigating a network of cellular decisions, each with consequences that evolve over time. The aging body is full of cells trying to choose between survival and self-destruction, between repair and retreat. p53 helps make those choices. Whether those choices lead to longevity or decay depends heavily on how well autophagy is working in the background. Understanding and preserving the p53 autophagy axis could prove critical to extending health span, delaying cancer, and preventing the chronic diseases of aging. But it must be done with nuance, recognizing that the very proteins that protect us from early death can, in a different context, accelerate decline. In this biological tightrope act, context is everything.